Welcome to our EOCT review module on cellular energy. Cells need energy to perform work and maintain a metabolism. During this module, you should complete the individual sections of your review packet. This will prepare you to participate in the vocabulary games following this demonstration. Key ideas included in this module are the structure and function of ATP, the summary equation for cellular respiration, the structure and function of the mitochondria, an overview of the stages of cellular respiration, the difference between fermentation and cellular respiration, the summary equation for photosynthesis, the structure and function of the chloroplast, an overview of the stages of photosynthesis, and the theory of endosymbiosis. Energy flows into an ecosystem as sunlight and leaves as heat. Photosynthesis generates oxygen and organic molecules, which are used in cellular respiration. Cells use chemical energy stored in organic molecules to regenerate ATP, which powers cellular work. This diagram illustrates that the processes of photosynthesis and cellular respiration are interdependent. That is, the products of one pathway serve as the reactions for the other pathway. The chemical potential energy temporarily stored in ATP drives most cellular work. ATP stands for adenosine triphosphate, a molecule characterized by having adenosine, ribose, and 3 negatively charged phosphate groups. The bonds between the negatively charged phosphate groups of ATP's tail can be broken down to release energy that can be used to perform cellular work. Once broken down, ATP becomes ADP, or adenosine diphosphate. This molecule has much less potential energy than ATP. A molecule of ATP can be regenerated by the addition of a phosphate group to ADP. Cellular respiration is the process that releases energy by breaking down food molecules in the presence of oxygen. All living organisms perform some type of cellular respiration, including autotrophs. Cellular respiration requires a food molecule, such as glucose, oxygen, and it gives off carbon dioxide. Because it occurs in the presence of oxygen, cellular respiration is also known as aerobic respiration. The chemical formula for cellular respiration is C6H12O6 plus 6O2 yields 6CO2 plus 6H2O plus energy. That is, glucose plus oxygen yields carbon dioxide, water, and energy. The reactants for cellular respiration are glucose and oxygen. The products for cellular respiration are carbon dioxide and water. Mitochondria are the site of cellular respiration in eukaryotic cells. Mitochondria have a double membrane that allows compartmentalization within the organelle that increases its overall efficiency. The outer membrane is smooth, but the inner membrane is highly convoluted, forming folds called cristae. Cristae contain enzymes important to ATP production, and the convoluted folds of the cristae increase the surface area available for ATP production. In the center of the mitochondria is the matrix, an aqueous environment where the chemical reactions of the Krebs cycle occur. The 3 main stages of cellular respiration are glycolysis, the Krebs cycle, or citric acid cycle, and electron transport. As is illustrated on the left side of the diagram, glycolysis takes place in the cytoplasm of the cell. During glycolysis, glucose is broken down into 2 molecules of pyruvic acid. Glycolysis produces 4 ATP, but because 2 ATP are used during the sugar splitting process, the net yield of ATP during glycolysis is 2 molecules. 2 NADH are also produced during glycolysis. These molecules carry electrons extracted from sugar to the electron transport chain. Notice in the center of the diagram that the Krebs cycle takes place within the mitochondrial matrix. In the presence of oxygen, the pyruvic acid produced during glycolysis passes into the mitochondria to the second stage of cellular respiration. The Krebs cycle completes the breakdown of glucose. During this cycle, pyruvic acid is broken down into carbon dioxide in a series of energy extracting reactions. Every time you exhale, you expel the CO2 produced by the Krebs cycle. The Krebs cycle produces 2 ATP molecules, 6 NADH molecules, and 2 FADH2 molecules. The NADH and FADH2 will shuttle their electrons to the electron transport chain to be used in the production of more ATP. Because citric acid is the first product of the Krebs cycle, this event is also known as the citric acid cycle. Following the Krebs cycle, the electrons captured by NADH and FADH2 are passed to the electron transport chain, where the majority of ATP is produced during cellular respiration. The electron transport chain uses the electrons carried by NADH and FADH2 to convert ADP to ATP. The energy of the electrons extracted from glucose is used to establish a concentration gradient of hydrogen ions across the inner membrane of the mitochondria. As a result of this charge difference, hydrogen ions escape through a channel protein called ATP synthase. Each time it rotates, the enzyme ATP synthase grabs a low-energy ADP molecule and attaches a phosphate, forming high-energy ATP. The electron transport chain is capable of producing approximately 32 to 34 ATP molecules, making the net yield of ATP produced during aerobic cellular respiration anywhere from 36 to 38 molecules. In the absence of oxygen, cells are still capable of producing small amounts of ATP. Fermentation and anaerobic respiration enable cells to continue to produce ATP even when oxygen is scarce. Two common types of fermentation are alcohol fermentation and lactic acid fermentation. Alcohol fermentation by yeast is used in brewing, winemaking, and baking. Lactic acid fermentation by some fungi and bacteria is used to make cheese and yogurt. Human muscle cells use lactic acid and fermentation to generate ATP when O2 is scarce during strenuous activity. It is important to note that while cellular respiration produces about 38 ATP molecules per glucose, fermentation produces only 2 ATP molecules per glucose. Photosynthesis is the process whereby light energy is converted to chemical energy stored in the bonds of organic compounds. In the presence of light, plants and other photosynthetic organisms transform carbon dioxide and water into simple sugars and release oxygen. The chemical formula for photosynthesis is 6CO2 plus 6H2O in the presence of light yields C6H12O6 plus 6O2. The reactants for photosynthesis are carbon dioxide, water, and the products are simple sugar and oxygen. Chloroplasts are chemical factories powered by the sun. They have a double outer membrane that creates a compartmentalized structure which supports its function. The structure allows cells to capture energy available in sunlight and convert it to chemical energy bonds via photosynthesis. Thylakoids are sac-like structures in chloroplasts made of photosynthetic membranes. The light reactions of photosynthesis occur in the thylakoid membranes. Stroma is an aqueous region outside the thylakoid membranes where the Calvin cycle occurs. Chlorophyll molecules are embedded in the thylakoid membranes. They capture energy from sunlight to power photosynthesis. The reactions that occur during photosynthesis can be broken into 2 stages. The light-dependent reactions, which take place in the thylakoid membranes. This is the photophase. Here, light energy is used to produce ATP and NADPH, both of which are required to power the Calvin cycle, and the light-independent reactions, also called the Calvin cycle. This takes place in the stroma of the chloroplast and is known as the synthesis phase. It uses the energy from the light reactions to fix carbon dioxide into simple sugars. Proposed by scientist Lynn Margulis, the hypothesis of endosymbiosis suggests that mitochondria and chloroplast were formerly small prokaryotes living within larger host cells. This means that eukaryotic cells evolved as prokaryotic cells began to form symbiotic living arrangements many years ago. Evidence for this hypothesis includes Both chloroplasts and mitochondria replicate by a splitting process similar to prokaryotes. Both chloroplast and mitochondria contain a single circular DNA molecule very similar to that of prokaryotes. And both chloroplast and mitochondria have their own ribosomes, which translate their own DNA into proteins. Now that you have completed this review module, log on to your virtual textbook and see how many of the vocabulary games your team can be the first to complete.